So also gives me great pleasure to introduce our next speaker, who is, I guess, what Buncha said, a rising star, Assistant Professor Diana Koh, and then he is an assistant professor at the National University of Singapore Cancer Science Institute, and he's also and he is going to be sharing with us about telomeres. If you don't know what they are, I'm sure he'll be very happy to tell you. And specifically, I think he'll be talking about sarcoma. So let's welcome Dennis, please.
Thanks a lot, Yvonne. Thanks for having me. As scientists, we love talking about our research in excruciating detail, but this presentation gives me even more pleasure trying to speak to a broader audience and also seeing what a wonderful impact the Diana Koh Foundation has already made, especially for the youngest scientists in our community. I was, however, a little self-conscious today speaking right after Buncha, who is a very accomplished clinician. Everything he does, right, he makes a real-world impact. People can relate to that. So the relief came in his title, emphasizing on research, and this resonated with me and reminded me of a ad campaign by the research-intensive German pharma companies that came up with that when I was starting as an undergraduate student, and it's running until today, and the slogan is, Forschung ist die beste Medizin, research is the best medicine. So as a first declaration, as it was said, I'm what you would call a basic fundamental scientist, and I'm going to share what we are focusing on in our scientific treasure hunt, hoping to make a small contribution to cancer therapy in the future. And so our reasoning starts with the notion that we all begin from a single cell. And then these cells have to divide many, many times. And it's estimated that a grown baby, adult human body later is made up of about 30 to 40 trillion cells. That's a lot of cell divisions, right? An enormous number. It becomes a little bit simpler if you remember the famous legend of the rice corn and the chessboard. There are different versions of it, but lo and behold, a king wanted to give a reward, and the rewardee just humbly asked for 1 rice corn on the first piece of the chessboard, 2 rice corns on the second, 4 on the third, 8 on the fourth, and so forth. And the king first thinks, oh, this is a humble request, I can easily fulfill it, until he realizes very quickly that the chessboard overflows of rice corns with very, very, very large numbers. And if we follow the pattern of the chessboard, we have to go maybe about 40 steps or so to reach our 30 to 40 trillion cells. That's more manageable, right? Like a cell divides 40 times. And that's exactly what happens. However, our normal cells at one point have to stop. They cannot do this forever. And the reason why they can't do this forever is embedded in our genome. So if you look at one of these cells, in this illustration, the center here is what we call the nucleus, where our genomic information, our DNA, is stored. And our DNA is organized in chromosomes. They have these characteristic egg-shaped structures, at least at some point during the cell cycle. And in the red part, we have the vital information, the blueprint that is a building block for making us as humans. There are 23 of these chromosomal pairs. They contain around about 20,000 genes, so again, the instructions of making the different cells in our bodies. And they're made up of about 3 billion letters of the DNA language, G, A, T, and C. And we have to preserve them at all costs. And we preserve them with these small blue caps surrounding them, which are made up of about 10,000 letters or so. And these blue caps, we call them telomeres. We can draw analogy to shoelaces for those of us who wear shoes with shoelaces and not sandals today. So we can think of a chromosome as a shoelace string, and they come typically with either plastic or metal caps that are meant to protect them. They're called aglets. And because we are all busy people, we run around a lot, we work hard in the research lab. With time, these plastic caps, they can fray out, They can break apart bit by bit until the shoelace completely opens up, and it's dysfunctional, right? And just like the shoelace, our chromosomes lose bit by bit their protective caps every time a cell divides until they become dysfunctional and the cell can no longer divide. First up, this has deep implications for the human aging process. It limits how often a cell can divide. If the whole process goes away and the telomere shortening is faster than in an average human being, it can lead to disease. Premature aging syndrome, such as progeria, where patients age faster. Basically, their biological clock runs at a higher speed. This is one of the very famous progeria patients, Sam Burns, who passed away about 10 years ago at the age of 17, and who was a strong advocate for the community of these patients. But also in a normal, average, healthy human being, this process takes place that the telomere shortens, and eventually it contributes to onset of gray hair. In addition to building our 30 trillion cells, we have to replace them. A very skewed ballpark number is that about 7 to 8 years, every single cell in our body gets replaced. And this cannot happen indefinitely, so we age. On the flip side, a fundamental hallmark of cancer cells is that they can divide forever. They can grow and grow at end. And so invariably, cancer cells have to find a way to keep their telomeres long enough so that they can grow and grow and grow. And so they activate mechanisms to do so. In addition, telomeres deal with a second problem. So we have these X-shaped structures, right? So there's a beginning and an end. And when something goes wrong with our DNA somewhere in the center where the genes are, where the blueprints are, there's very proficient repair mechanisms to patch things up and to restore our chromosome, right? Again, we have to preserve that information at all costs. There's very extensive research from groups like Ashok's that have elucidated these key mechanisms. Essentially, telomeres are permanently a problem. They're permanently broken. And if the same repair machinery would recognize them as such, then chromosomes will fuse to each other. This is a real microscopy image of human chromosomes. We can somewhat recognize in blue these X-shaped structures. We mark the telomeres here in red, and many of them are stuck to each other now. And that is incompatible with life. Now, this shortening with every cell division that I introduced is one way how we can end up with a situation that there is no protection anymore and chromosomes now fuse to each other. How do cancer cells, you know, as scientists, we like graphs, so bear with me for a second. I try to break this down. So we've now seen that we maybe start with long telomeres at the beginning of life. Cells divide and divide and divide, and they shorten. And at one point, most cells reach a stage where they stop dividing. They senesce. They contribute to aging. Most of them will die eventually. Some cells might now have already acquired mutations along the way that allow them to keep going a little further. The telomeres get even shorter, and they end up in this situation. Again, most cells will die here. But some of them will somehow manage to rearrange the DNA in a way that they escape what we call the crisis, and they can keep going again. And at that moment, they will turn on also mechanisms that will, again, maintain their telomeres and make them longer again. And there are two of them. The first one is an enzyme called Telomerase that is found in about 90% of all cancers. It helps to go back to the state, and the cancer cells can keep growing. 90% of cancer is fantastic, right? It makes it a really attractive target for therapy, and naturally, companies have developed inhibitors for this enzyme, such as imetelstat or Ritello. And those are in clinical trials, and there's promise. There's still a lot of work going into it. But one challenge with this type of approach is that we might turn off the telomere maintenance, and then the cancer cell can still divide for a while before the therapy really kicks in. This is in parts also true in the remaining 10% of cancers, but there we have another opportunity. The remaining 10% of cancers use a mechanism that is called alternative lengthening of telomeres, or ALT for short, with the idea that one chromosome, one telomere, copies itself from another. And if we were just to turn off that mechanism, we would eventually run into the same problem. Now, this sort of mechanism is particularly prevalent in some cancers, at 10% overall, but it's not equally distributed across all cancer types. And it is particularly prevalent in gliomas and sarcomas. And while there's variation between cancer types and treatment options, overall, oftentimes, patients that are ALT positive respond more poorly to therapy. Now, as I said, if we just turn off the mechanism, this might be one way to do it, but maybe the effect would kick in relatively late, depending on how soon we find the cancer. But the ALT mechanism gives us a second opportunity. This actually does not exist ever in any normal cell in our body. So we can also think about going the other direction and activating it even more. That should have relatively little side effects because it does not exist in normal cells. But it could push the cancer cells over the edge so that now they try to copy themselves so much from each other that they get entangled with each other and they are stuck. And I just want to showcase one such examples from one research project where we have managed to push the cancer cells over the edge in this way. Here we take a protein that is fused with a fluorescent marker that allows us to visualize it under a live cell imaging microscope, so it's in green. And you can see these individual dots here that actually demark the telomeres inside of the cellular nucleus that lights up here. And we follow these cells as they divide. Up here is a cell that will now split into two. And usually cells split into two and they're nicely separated and they go on and they keep doing this. But these cells, they have this sort of tether, this kind of umbilical cord between them, which represent the telomeres that are now stuck to each other. And these cells try to get away from each other. They pull and pull and pull, but they can't, and eventually they end up dying. So we hope to exploit this mechanism in this way to turn it on even more, to basically turn the cancer against itself in order to selectively kill it. And this idea is not uniquely ours. It's an emerging concept in the field. In fact, there are already some startup companies that think along these lines and that are starting to develop drugs to exploit ALT-positive cancers and to selectively kill them in this fashion. Now, as cancer scientists, oftentimes we have two options. We can dive really, really deep into these mechanisms, figure out one thing, and then think about how we can tinker it and use it. But we can also try to take a hawk's eye view. As I said earlier, our genome is very complex. There's 20,000 genes. Those can make 20,000 proteins and actually even more. And so we might overlook very, very meaningful information. And so the second approach that we take is to try to look at everything systematically and then to see what the data tells us. And I just want to give you one brief glimpse of that, where we have collaborated with clinicians at NCIS, at NUH, to look at a cohort of 75 sarcoma patients, and then we have a molecular assay to read out whether they are ALT positive or not. These cancers, they make sort of circular DNA product. Suffices to say, we have this assay where we spot the DNA on a membrane, and if you see a dark band, they are positive for this feature. And out of some of the patients that are spotted here, patients 32, 34, 37, 41, they're clearly positive for this. And then we looked at all of them, and about a third of the Singaporean sarcoma patients are ALT positive. So this is a prevalent feature in the local patient community. And this might be actually one of the first systematic data sets that have looked at this in this region. And we can now take these cancers and use approaches that will look at all the proteins in parallel and ask what might be different, what might be special to these kind of cancer patients. In our lab in particular, we use a set of instruments that are called mass spectrometers. We can think of those as molecular balances that weigh molecules, and this way we can distinguish all these 20,000 different proteins, and not only distinguish them, but also ask how much of each is present, to then look at proteins that might be uniquely abundant in the cancers that are positive for this ALT feature, or that are particularly devoid in this feature, to have a wider net to find new either diagnostic or therapeutic targets in the future. All right, I want to stop at this point and highlight that obviously it takes a village. It already takes a village within a research group, so I'm super fortunate to work with an amazing bunch of people, but it also takes a village in the wider sense. And so the last few slides were also an example where we had the fantastic opportunity to work with our clinical colleagues, such as Anand and Victor at NSYS, literally to the point where we have one program set up where on the day of surgical resection, the tumor comes into the lab and is prepped immediately for these kind of analysis, and that obviously has a whole string of requirements from ethical considerations, right, from logistics, until we can do the actual research. And in Singapore in general, I think we're also blessed with a very science-friendly environment and generous funding, and today obviously is a great example of that spirit. So on that note, I'd like to thank all of you, and I'm happy to take any questions.
Dennis, questions for Dennis? Leah, you sure?
Hi, thank you for your talk. So I'm curious to know, regarding the telomerase, does the cancer only rely on the telomerase early on when converting from the normal to the cancer type, or do they also depend on it as the cancer develops like several years later?
Yeah, they continuously depend on it. So they at first have to get to a stage where their telomeres are long enough again to keep growing, and then they perpetually depend on it, because if you take it away, they would then go down the fate of a normal cell again, that they can't divide enough. The challenge here is, when do you take it away, right? If the tumor is already huge and you take it away, maybe it has these cells have enough divisions in them so that the tumor wins the fight. If you were to take it away maybe very early on, then maybe you stop the growth when it's still very meaningful for the patient. So that's sort of like the tricky part about it.
Thank you.
Thank you. Thank you, Dennis, for the really nice talk. So I would like to ask about the lifestyle imaging video that you showed just now. May I know how long was the experiment? Because maybe I missed it, but the cells eventually die because it fails. Oh, okay, so it was really a very long experiment. Because actually I have seen a paper whereby some cells, they did fail to divide, and because of that, it cannot divide, right? But at the end was that the cells went through reverse cytokinesis. So the two cells that failed to divide came back together, attached again, and they just don't divide, but they survive.
Yeah, so the different ways to introduce this, the different genetic manipulations that we can do here to just sort of activate the pathway and then have fusions. And clearly also these cells, they have other alterations, right, so that they're not stopped even before they try dividing. And then once they divide it, they will not go back. Actually, they have completed the cell division apart from this thread of DNA that is entangled. And they try to get out of it, but eventually, because they can't truly complete, right, and then they can't go to the next cell cycle. So they'll essentially either just stuck or die from that situation.
Thank you for sharing. I just curious for last two slides share about the Singapore cohort is only around less than 100 patients. So because sarcoma is a group of patients origin from different area. So I don't know how about the other cohorts and also whether this patient is age different because trauma is also related to aging, right?
Yeah.
Thanks.
Thank you very much for this. So sarcoma is a relatively rare cancer compared to other cancer entities. That in part explains why the cohort does not have 1,000 samples straight away. And so it's the beginning. We want to grow this because there are many different subtypes, right? So we would want to look at the same question in only liposarcoma patients, in only osteosarcoma patients. And so it takes time to gather these samples. One way how we also approach this is to then start collaborating with other hospitals internationally. That also that gives us sort of the chance to have an independent set of samples. Oftentimes it's better to look in 2 times 100 patients that are totally independent than 1 times 200. But also it gives us a chance to compare, right? It's a difference in sarcoma patients that have a Caucasian, Eurasian ethnic background compared to patients that are from Southeast Asia. Yeah.
Wonderful presentation. So why do some cancers choose to go down one path using telomerase mechanism versus the ALT mechanism?
Thank you, Anthony. That's a key research question in our labs that we want to understand. Actually, for the ALT pathway, it's not truly known how it's activated. We know parts of it, but not the full story. The one thing that I can say is that we have different lineages in our body when we develop, and this ALT pathway is particularly prevalent in the mesenchymal lineage that gives rise to fat tissues, that gives rise to bone, and so on. So there's something there, but we simply don't know yet. Yeah.
Do you use your research on patients in real time for treatment? I want to see the link between your research and treatment.
Yeah, in real time, not yet. That's sort of like one of our pipe dreams. So we have this platform where we can, together with the surgical team, get the patient samples into the lab on the same day. And also with these last slides where we can look at many factors at the same time. In the future, we'd love to see a setup where, let's say, within a week or 2, we have completed our analysis, and then we can sit down with the clinicians again and look at how this data can immediately benefit this particular patient. We aren't quite there yet. At the moment, it's still a slower process, and we look more retrospectively, but that's exactly the direction where we're headed.
Thank you for the great talk. So I was wondering, is the rate of telomere shortening tissue-specific?
That's a great question. I don't know if I have a very good answer. I think I would actually have to look through literature a little bit. I would believe so, but also one confounding factor is the rate of proliferation across tissues is very different, right? So blood, for example, is extremely heavily studied because it's so accessible, and that's probably the tissue where you would see the decline the fastest, and that's also typically what's used to, for instance, identify individuals that have particularly short telomeres that are prone to some sort of either cancer or aging syndromes. Yeah.
So thank you everyone for participating. Thank you, Dennis, for a really inspiring talk.